In the previous class, we have discussed about the discovery of the restriction enzymes. In this class, we will learn much more details about the restriction enzymes. We will start with the major class, which is nucleases. Nucleases are the enzymes which break nucleic acids. Now, nucleases are divided into two types. The first type is exonucleases. Exonucleases are the enzymes which break the DNA at the ends. So here you can see these enzymes are breaking the DNA at the ends. So after breaking, you can observe something like this. So they are breaking the DNA at the ends. Endonucleases. So these are the second type of the nucleases. They break the DNA from the middle. They break the DNA from the middle point. So these endonucleases are the enzymes which recognize specific recognition sequence and break the DNA from the middle, or they cut within the DNA. Such kind of enzymes are known as endonucleases. Endonucleases are again of three types: type 1, type 2, type 3. But in the rDNA technology, we use type 2 restriction endonucleases. So what you have to remember is endonucleases are considered as molecular scissors or biological scissors. Restriction endonucleases are the enzymes which we are using in the rDNA technology, and these are specifically recognized as restriction enzymes, molecular scissors, and they are again of three types: type 1, type 2, type 3. In that we use type 2 type of the endonucleases majorly in the rDNA technology. Now let's understand how the mode of action of the endonucleases will be. Right. So restriction endonucleases, two important things which we have to remember. Number one, these enzymes recognize a palindromic sequence. Now what is a palindromic sequence? It is a sequence which can be read same in either direction. So that means what? Let's take an example. Imagine Malayalam. Now Malayalam is a palindromic sequence. So here in this forward direction, if you can read, it is the same kind of letters which you can notice. And even backward direction also you can read the same kind of letters we can notice here. M-A-L-A-Y-A-L-A-M. So such kind of sequence which can be read same in either direction is known as a palindromic sequence. Now restriction endonucleases. First one is it recognizes palindromic nucleotide sequence. So a palindromic nucleotide sequence it recognizes. Number one. Number two, there is a specific recognition sequence for specific restriction endonucleases. Now that we will understand by taking some examples. Now we will start with the first type of the restriction endonuclease, which is Echo R1. So Echo R1 is a type of the restriction endonuclease. It is a restriction enzyme. Right. Now this echo R1, it recognizes a palindromic nucleotide sequence. Right. Now what is that palindromic nucleotide sequence is? GAATTC. Right. From 5 prime to 3 prime, if you can observe the nucleotide sequence GAATTC. Now again, here from 5 prime to 3 prime, we can observe that what is the nucleotide base sequence in the other strand? GAATTC. So we consider this sequence as palindromic sequence. ఇక్కడ palindromic sequence అంటే 5 prime to 3 prime. What is the sequence? GAATTC. ఒక strand మీద 5 prime to 3 prime ఉన్న sequence GAATTC. Other strand, this is a DNA is a double stranded right here. So in the other strand, if you can notice 5 prime to 3 prime, again it is the same sequence GAATTC. So that is why this DNA segment which you are seeing here is a palindromic sequence. 5 prime to 3 prime. In both the strands, if you read, we get the same nucleotide sequence. So echo R1 is one type of a restriction endonuclease. Now this echo R1, first one is it recognizes a palindromic sequence. What is that palindromic nucleotide sequence is? GAATTC. From 5 prime to the 3 prime in both the strands. Now, right. The second thing is echo R1 has a specific recognition sequence. What is that specific recognition sequence is? GAATTC from 5 prime to 3 prime in both the strands. So that is the recognition sequence of echo R1. Right. Now what is its action? Now let's understand. Now the echo R1, when it identifies this recognition sequence, right, it cuts between G and A. So this is a 5 prime to 3 prime. So you can observe here it is cutting between G and A. So when it cuts the DNA between the G and A, so here a PD bond has broken. A phosphodiester bond has broken. And in the other strand also, 5 prime to 3 prime, GAATTC is there. So it cuts between G and A. So here is a cut in the other strand. Right? Now here also a phosphodiester bond has broken. A PD bond has broken. So here a PD bond broken in the strand 5 prime to 3 prime, and here also there is a PD bond broken. So 2 phosphodiester bonds have been broken. Now what is happening is, because of this uneven cut, this is known as an uneven cut or a staggered cut. So because of this uneven cut or staggered cut, what happened is the hydrogen bonds also became loose, because the 2 strands have become loose. So what happens? The hydrogen bonds have broken automatically. So right here you can observe the hydrogen bonds are also broken. So here this is the cleavage. So the hydrogen bonds are also broken. 8 hydrogen bonds are broken. 2, 2, 2 and 2. So totally 8 hydrogen bonds broken. 2 phosphodiester bonds broken. And this cut is an uneven cut or a staggered cut. So after breaking, we are getting 2 fragments here.